one of... Hi, we're Group E, Kane, Carrie, Connor, Callie, and Maya. Today, we will be discussing the question, why do stem cells remain stem cells? But before we go any further, we're going to have to give a quick background on stem cells. Stem cells are unspecialized cells, and they have the capability of dividing even after long periods of dormancy. In addition, they can also be induced to develop into tissue or organ cells that are capable of specialized functions.
Let's talk about plants. Life is dependent on the continual growth of plants. The growth of plants is dependent upon the specialization of plant meristems, undifferentiated cells found at the tips of shoots and roots, that can become any type of cell needed by the plant. However, just as important is the stagnancy of meristems. Plants do not waste energy specializing cells when deemed unnecessary or unhelpful for survival. So what keeps these meristems and not differentiated cells?
Now let's talk about animals. There are two sources of stem cells found in animals. The first source is in embryos formed during the blastocyte phase of development, and these cells are referred to as embryonic stem cells. Embryonic stem cells continue to divide and remain undifferentiated until a chemical signal is received telling them which cell they will develop into. The second source is adult tissue, which produces adult stem cells. Adult stem cells exist all throughout the body and can be found in different types of tissues such as bone marrow, blood vessels, and skin. These cells remain undifferentiated and non-dividing for years until they are activated by tissue injury or disease. Once the cells are activated, they can divide indefinitely and replace the tissues of the originating organ.
The main culprit behind the undifferentiating meristems in plant cells was found by Dr. Thomas Laux at the University of Freiburg. He indicated that special microRNA found in only the epidermal layer of the plant shoot tips prevented specialization. This is unlike other plant microRNA found in other cell layers that do indeed initiate cell specialization. Since the microRNA that prevents specialization, known as the stem cell preserver, is not found in other cell layers of the plant, those cells are free to differentiate to whatever they need to become. The part of the epidermal layer to which the stem cell preservers are located contain unspecialized cells because of the abundance of such microRNA. These stem cell preservers prevent proteins from being generated by other RNAs. As the stem cell preserver finds its way to other cells, the freed cells are now able to receive specialization-initiating microRNA that help them differentiate as needed since the proteins can now be derived.
In addition to the primary article, another article was found that describes a transcription factor that can be targeted in studying shoot apical meristem, otherwise known as SAM, a good source of studying plant stem cell homeostasis. Stem cell fate in SAM is controlled by the transcription factor WUSCHL or WUS. However, understanding of cell-to-cell communication and control of cell behavior within the stem cell system is still fairly limited. The scientists in this article showed that the BHLH transcription factor HEC1 or HEC1 is the target of WUS. It also contributes to SAM function by promoting stem cell proliferation and also counteracts niche cell activity. HEC1 controls genes with functions in metabolism and hormone signaling. The targets of these transcription factors are phytohormone response regulators, which are shown as mobile signals. Their work shines light on mechanisms that guide meristem function and show that the overall system is more complex than previously suggested.
Animal stem cells must be able to move through micron-sized pores to reach their intended location. This trait is similar to how cancer cells move throughout the body, causing scientists to study this in hopes of understanding cancer cells better. Due to the pores being so small, the nuclear membrane often cannot pass through the pore. Lamin A is a nuclear protein which determines the stiffness of the nuclear membrane. High levels of lamin A result in a stiffer membrane, hindering its ability to move. Though, if levels are too low, membranes have the possibility of breaking. Scientists have also discovered that extremely low levels inhibited the cell's ability to react to changes in heat and pressure, leading to increased chances that the cell would die.
Research has also been conducted to determine why stem cells begin to differentiate into their desired cell type. A research team led by Dr. Shulamit Levenberg studied how the differentiation and division of a human embryonic cell can be promoted using biodegradable polymer scaffolds that provide structure and support for the cells as they grow. Their research found a positive correlation between the polymer scaffolds and the strength and desired differentiation of the cells. While they found that the signals required for the differentiation to occur were present, they also found that structural support such as PLLA or PLGA polymer scaffolds were necessary to induce the growth of a 3D tissue structure. The team also found that when a polymer scaffold was present, higher amounts of the differentiation chemicals cytokeratin and nestin were found in the tissue. These suggested that the polymer scaffold was successful in aiding the differentiation of the tissue cells, since a high correlation between these chemicals and the formation of epithelial tube structures is known to exist.
Let's discuss why meristems stay meristems. Plant meristems stay plant meristems due to the special microRNA that prevents the creation of proteins by other RNAs in stem cells that leads to specialization. These microRNA are vital in the plant's attempts to presumably save energy, but at the same time grow when needed for survival. In addition, Lox's experiment shows the importance of microRNA in both differentiation and non-differentiation of meristems. Simply put, without them, the plant could not grow, and thus life on Earth would be severely altered.
Overall, it was found that HEC1 is the target of WUS, which contributes and promotes stem cell proliferation and also controls genes that function as metabolizers and hormone signalers. This shows that blocking these transcription factors will not lead to stem cell proliferation, and microRNAs are a way to prevent transcription factors from actually transcribing stem cells to proliferate and specialize. This shows how stem cells remain stem cells instead of growing into various tissues or organs with specialized functions.
Now let's discuss how animal stem cells differentiate where needed. Stem cells are stored in most organs of the body and are specific to that organ. Once they receive a signal to begin the differentiation process, they must move to the area that new cells are needed. We learned earlier that the cell's level of lamin A determines its ability to move through small pores to reach its intended location. Once they arrive, high levels of determining chemicals decide what type of cell it will become. These chemicals are specific to each organ in adult stem cells, but almost all determining chemicals are present in embryonic stem cells.
In conclusion, this knowledge of plant and animal stem cell differentiation is important not only to know how they work inside our body in plants, but also so that scientists and researchers could continue their advancements in medical treatments that save lives. Thank you!